my introduction all that much. It's not what they came to see. So today it's my pleasure to introduce Dr. Jacqueline Crawley, who will be sharing her work on behavioral phenotyping of genetic mouse models of autism. Dr. Crawley earned her Bachelor of Arts at the University of Pennsylvania before earning her PhD in zoology at the University of Maryland. Dr. Crawley's postdoctoral work was in neuropsychopharmacology at Yale, and over her career, she's had a strong research career for a long time at the NIH with a short stint, it looked like, at DuPont, and adjunct positions with the University of North Carolina and Georgetown University, before moving to her current position where she holds the Robert E. Chasen Endowed Chair in Translational Research at the Minds Institute at the University of California, Davis. And Anna reminded me that she, I think, was a previous IBANGS president and a distinguished research fellow. I'm not looking at that email, so that's off my memory. So with no further ado, I'd like to thank Jacqueline for agreeing to give these seminars. They're turning out to be a great sort of presence for IBANGS, and I'm glad people are all involved in putting this together, and can't wait to see what you have to say today. Thank you.
Thank you, Carl, for the kind introduction, and I'm so grateful to Paul Meyer for inviting me to give this talk to IBANGS, our great society, and all of the members who are here with us today. So I'm going to go ahead and start sharing the screen now, hopefully. That's showing up okay?
That looks great, Jackie.
Perfect. Okay, thank you. So we're going to be talking today about mouse models of autism and how one conducts behavioral phenotyping, and how our lab is using the models with the strongest phenotypes to look for therapeutic interventions based on the pharmacological outcomes of the genetic mutations that have been identified. So to start off with, some of you may not be so familiar with autism spectrum disorder. This is a neurodevelopmental disorder, usually diagnosed about ages 2 to 5 years old. The prevalence is remarkably high, greater than 1% in the population, independent of ethnic or geographical location. The improved diagnosis is the primary reason for the apparent increase in the prevalence of autism. We didn't even know about this disorder. It wasn't even identified as a separate diagnosis until the late 1990s. Vaccines do not cause autism. Let's put that to rest right from the start. And the diagnosis is defined behaviorally, which I'll tell you a little bit about. Of the many proposed causes of autism spectrum disorder, the strongest evidence so far is genetic, which is why this is appropriate for an IBANGS talk today. There are multiple causes of autism in the genetics domain. We originally understood that there should be a genetic basis by the initial studies on the monozygotic twins showing a very high concordance, 60 to 90%. If one had autism, the other identical twin would have autism, versus 10 to 20% in dizygotic fraternal twins or siblings. So that differential really indicated we should be looking at the genome. The frequency of boys to girls is also another indicator. It's at least 4 to 1 times more common in boys than girls. So there, as you can imagine, there have been many huge international consortia searching for genes linked to autism, and association studies and whole genome sequencing have identified chromosomal deletions and duplications, including at chromosome 15, 16, 22, and the X chromosome. There have also been epigenetic factors identified, including chromatin remodeling genes, such as CHD8 and MECP2. There are multiple putative risk genes. Each person with autism might have one of these genes, and we're now up to over 100, probably over 200 potential risk individual single mutation, de novo risk mutation, gene mutations. Many of these are in synaptic proteins, such as the shanks, neurexins, neuroligands, and contactin-associated proteins. Many are in neurotransmitter-related genes, such as the GABA beta-3 subunit of the GABA-A receptor, the serotonin transporter, the vasopressin 1A receptor. Some are in developmental genes, including some that are also risk genes for cancers, including P10 and MET and GRAIL2 and WDFY3. And we also have comorbidities with other genes implicated in other neurodevelopmental disorders, such as Fragile X syndrome, Rett syndrome, tuberous sclerosis. This is intellectual disability, motor dysfunctions, seizure disorders. And there's quite a few environmental risk factors that may contribute a small percentage to risk for autism spectrum disorder. These are mostly still under investigation. They include older parental age, usually fathers over age 50, immune dysfunctions, maternal autoantibodies, cytokines mediating early brain development, and some interest in environmental toxins. So what is autism, in fact? It's definitely a heterogeneous disorder. The two main diagnostic categories are shown here. The first focuses on unusual reciprocal social interactions. So for example, lack of eye contact, looking into each other's eyes when in contact with other people. Lack of joint attention. So usually if one person points at a bird, the other will look at it, and often people with autism just go on playing with their toy instead. Interpreting facial cues or social cues is definitely a problem, to know that this person is happy and laughing and this person is not happy and angry. Then deficits in social communication are also the major limitation to people with autism developing good social networks and being able to hold jobs. These include delayed development of language, very concrete use of words, missing all the nuances and sarcasm and jokes and other modifiers of the actual concrete word, speaking in monotones, lack of prosody, lack of interactive conversation, more monologue kind of speaking. And mentioned the inability to interpret facial expressions, also body language, which is a big part of our understanding of what people are telling us. Inability to understand what other people are really thinking and feeling and intending, which is again necessary for normal social behaviors. This has been called theory of mind or mentalization of mind blindness. The second diagnostic category includes a group of major repetitive behaviors, motor stereotypies, doing one thing over and over again, insistence on sameness, being very, very upset with any change in routine in meals, TV shows, driving routes, which is a big problem for the families if they want to go on vacation or do anything unusual outside of the routine. Often people with autism are really into one thing. They have a very narrow, restricted interest in something like baseball statistics or bus routes or collecting rocks or what the radio stations are, elements of the periodic table. Sometimes this leads to really remarkable special abilities, such as in math or in music or in art, which in some cases have been led to call people with autism savants, which may simply just be that they're really good at one thing. But allows many people with autism to hold jobs and have very interesting careers. There's also associated symptoms of autism that don't meet the diagnostic criteria. They're not found in everybody, but are fairly common. Intellectual impairment is very common. Maybe almost up to half of people with autism have low IQs. Seizures in about a third of people with autism. Hypersensitivity to specific sensory odors or textures or tastes is very common. Hyposensitivity to others, such as painful stimuli, have also been reported in some people with autism. Anxiety is very high. Up to 50% of people with autism meet the diagnostic criteria for an anxiety disorder. Many have ADHD as well, and a lot have sleep disruption, which, as you can imagine, is really hard on the family, especially when they're small children. Gastrointestinal disruption has also been reported. So again, this is a heterogeneous disorder. The severity of the symptoms really varies across individuals, and the associated symptoms are present in some people and not in others. So they say if you've seen one person with autism, you've seen one person with autism. And that's a real challenge for understanding the genetics and for developing therapeutics. There may be very clear subgroups and very diverse subtypes to consider. So as mentioned, there's been mutations in hundreds of genes that have been implicated in autism. And mouse models have been generated to address the consequences of many of these mutations. And many of you here at IBANGS were involved in the development and the application of some of these mouse models, including through the International Knockout Mouse Project. So we now have a very large number of mouse models for many of these human risk genes. These are just a few. Some of them are these comorbid syndromes. Some of them are, again, receptor subunits, such as the GABA beta 3 subunit. And so we've got good models. They're available through JAX and other repositories and from our collaborators. So we began back in the early 2000s asking this very fundamental question: how do you model these human behavioral symptoms of autism in mice? So if you're a graduate student or a postdoc looking for a project and you want something with a real intellectual challenge, this was one that caught my eye immediately. The field really needed good behavioral people to think about what mice do that might be analogous to the human symptoms. And of course, we don't anthropomorphize. This is a uniquely human disorder. We never use this phrase in the lab. Nobody's allowed to use those two words together. What we're generating are behavioral assays with conceptual similarities to the symptoms. That's called face validity. And based on hypotheses about causes, such as genes or paternal age or autoantibodies, that's called construct validity. And then if you have a good mouse model or rat model or primate, non-human primate model with good construct validity and good face validity, that becomes a translational tool to use in various ways, including looking for treatments. And that's called predictive validity. So how do you get started? Again, if this was your project, somebody handed it to you, what would you do first? Well, what we did first was go to the clinical experts to really understand what these symptoms are. And we're very grateful to some of these fabulous clinicians who know all there is to know about the symptoms of autism at this point, who've let us come in and watch their videos and sometimes with permission watch diagnostic interviews through a one-way mirror and come to the INSAR, Autism Society for Autism Research meetings, and really learn about what the actual symptoms look like and their variability. We've gotten mouse models from our collaborators and from JAX. We conduct primarily the behavioral phenotyping across developmental stages and some of the assays for biological mechanism, and finish up with the drug treatments that we've been initiating in recent years. So these are some of the mouse models that we've been testing over the past few years. I'll be focusing today on the SHANK3B in GRAIL2 16p11.2 and an inbred strain called BTVR. So this is what mice do. When you put two unfamiliar mice together, they follow each other. They do a lot of nose-to-nose sniffing, nose-to-anogenital sniffing, groom each other, push past and climb over and under each other with a lot of physical contact. And so these are discrete parameters that are straightforward to score. So you put two mice together and there's the physical contact and there's the following. And we use video tracking or event reporting software. We've been using the Naldus systems, and other people use Cleversys, and some of you have developed independent, very user-friendly and open access scoring systems that are available online. So for example, here's the SHANK3B male mouse interacting with an estrus B6 female, following much less in the knockouts as compared to the wild-type litter mates, showing less total sniffing time of various body regions, and engaging in less physical social contact in the knockouts versus the wild-type. Well, this is very laborious, as any of you who have scored with Naldus Observer and from straight from videos understand. And we wanted to get a higher throughput method in place that would be simpler and automated. And we again talked to the experts and learned that children with autism tend to play with their favorite toy or get engaged in their special interest and not approach other children and not join in games with another kid. So what we developed was a 3-chambered system analogous to this phenomenon seen in the clinic and in the real world. We developed this in collaboration with Cheryl Moy and Joe Piven at University of North Carolina back in 2003, and Jacqueline N. This is a 3-chamber test that's now pretty widely used. You'll see it in a lot of publications on mouse models of autism. So there's photocells across the entrances between chambers, where this can be done with video tracking. This is just inverted wire pencil cup that mice like to sniff and crawl around on. That serves as a novel object, and it's just weighted down with a cup on top with a lead weight to keep the mouse from spending its time climbing on top. On the other side is the identical cup with a novel mouse inside. You could put a novel object in here. It turns out that doesn't really matter. The novel object is the wire cup. But here we have a novel mouse, which adds the social component. And so if you put the subject in the center chamber, give them the habituation time first, and then allow a 10-minute sociability session, the subject will spend more time over here with the novel mouse, exploring and sniffing, than over here with the novel object. This is a simple yes or no definition of sociability. More time with the novel mouse than with the novel object. This is the BTBR inbred strain, not a mutant line, just available, that we discovered through a strain distribution study through JAX. And here's the BTBR showing normal physical and social sensory and motor abilities, able to walk around, no problem, but spending about the same amount of time with the novel object as with the novel mouse. And that's the absence of sociability, again, by the simple binary yes or no definition. And so what the data look like for a standard B6 inbred strain, spending more time with the novel mouse in red than with the novel object in green, and the BTBR showing about equal time with the novel mouse and the novel object. We also record the center time as a control for just general locomotor activity, which could introduce a confound if it's unusually low or unusually high. Luckily, if you score the actual social interaction by quantifying time spent sniffing, over here, we get a good correlation between sniffing and time in the chamber with the novel mouse. And so that correlation gives us confidence that the single automated measure of time in the chamber may be sufficient. But with the new strain, we always look at actual social interaction through the sniff parameter to confirm that we're not studying an artifact such as the mouse just sitting in the corner in the same chamber as the other novel mouse. And here's how the sniff time data would look: more time with the novel mouse than with the novel object, and the same more time sniffing the novel, not sniffing, the BTBR not spending more time, significantly more time with sniffing the novel mouse than the novel object. We do not compare across genotypes or across inbred strains or across treatment groups. A lot of people use an index of sociability or compare these two bars. In our experience, this is just too variable a task for that kind of quantitative comparison across groups. We only use a yes or no within group, and if there's sociability or not sociability, that's the outcome measure. If you really want to be quantitative, you want to get back into scoring individual parameters from videos with a reciprocal social interaction task. Just wanted to be clear about that because you'll see a lot of over-interpretations of this task in the literature. So here's an example from the INGRAIL2. Again, this is the developmental gene regulating hindbrain and midbrain development early in embryonic development. And this is the work of our former postdoc, Jennifer Brailmeyer, showing more time with the novel mouse than with the novel object in the wild type and the heterozygotes, but not in the null mutants. That's on chamber time, same thing on sniff time, lack of sociability in the null mutants. And we also score a number of entries between compartments as, again, another control for general locomotor exploratory activity, and that was not different across genotypes. We wanted to make sure that this was, in fact, replicable, reproducible finding. And so Jennifer ran a second cohort up in GRAIL 2 and found the same pattern, good sociability in the wild type and hets, but not in the null mutants, for chamber time and for sniff time, and no genotype differences across exploration. I think this is during the habituation phase. And I'm showing this cohort 2 to make the point that you really do want to generate a whole nother second cohort and replicate findings when you're first studying a new mutant mouse model of autism. Just as in any area of science, you want to replicate your findings, but especially with behavioral studies in new mouse models, this turns out to be essential. There have been a lot of publications showing social deficits in a mouse model of autism with a new line of mutants that turned out not to replicate across other labs or even within the same lab. So please, again, if you're a student thinking about how you're going to design your experiment, even though it's expensive and it takes more time, try to design in a replication study. Social communication, as you can imagine, is difficult to model in mice, and the deficits are mostly in the language domain in autism. What mice do is they emit social pheromones, olfactory pheromones, for most of their social communication. They also emit vocalizations in the ultrasonic range, which we now have very good equipment to dial down to our hearing range and software to count number of vocalizations and properties of calls. So here's the olfactory test we've been using. Just rub a cotton swab, dip it into different odors, social odors, non-social odors, and count time spent sniffing a sequence of odors, both social and non-social. And here's how we look at vocalizations. Here's a soundproof chamber, foam lined, with a video camera that's recording the social session. And here's the ultrasonic microphone. We use one from a company called Avisoft that has very good software and also very good support for problems you might run into to get numbers of calls and various parameters of call frequency, and I'll tell you a little bit about types of calls. So if you put a male mouse into a chamber with urine on the cotton swab from a female mouse in estrus, here's the tracings that you'll get from the software, and here's what it sounds like if you lower this down to our hearing range. Can you hear that? Is that audible to you?
Yes, yes, Jackie, it's audible.
Beautiful, glad you could hear that. I don't know what that sounds like to you, but to us, it sort of replicates some of the properties of birds chirping.
Yeah.
Or if you speed up whale calls, it sort of sounds like vocalizations you might get from whales, the point being that it involves a lot of complexity. And our amazing postdoc, Maria Luisa Scattoni, looked at that complexity in great detail and was the first to show that there are these categories of calls that she could identify as short, long, frequency step. She was able to label 10 different call types that varied across wild type versus knockout mouse models of autism, and was unusual in inbred strains such as BTBR. So although the playback studies have not been done to definitively characterize communication in adult mice, we have some interest in the possibility that these do have syllable-like communication functions that could be exploited to look at the communication domain in mouse models of autism. But just in terms of number of calls, here's the C57 Black 6J, which does a lot of social interaction, and it emits a lot of vocalizations in the ultrasonic range during those interactions. And here's the BTBR mouse, which engages in fewer social interactions and emits very few calls during social interactions. Same thing with the Shank 3Bs, lots of calls in the wild type littermates and very few calls in the knockouts. Okay, repetitive behaviors in mice are a lot easier to model. Mice show spontaneous stereotypies. a lot of repetitive behaviors that could be linked, shown in self-grooming, in jigging, in circling. For example, here's the BTBR that shows very high levels of repetitive self-grooming. This is the normal pattern of grooming, so it's not a stereotype behavior, but once the mouse gets started, it grooms and grooms and grooms. Instead of just a few seconds for the kind of housekeeping grooming that you normally see in mice, this can go on for 30 seconds, maybe as long as a minute. Similarly, the Shank 3B mouse has a very high self-grooming score, replicated by almost every lab that studied this mouse. Again, it's the normal pattern. It's just very long durations of self-grooming bouts. As you know, we have, from all of you who work in the field, we have very good behavioral assays that are available in mice for some of the associated symptoms of autism. And we try to look at all of these when we do our very first characterization of a new line of mouse. And some of them do show some of these, and many don't. So open field is a good first analysis for motor functions, and also can pick up components that might be analogous to ADHD, high levels of locomotion. We have anxiety-related tests. This is the light-dark test that our lab developed back in the early 1980s, in which mice emerge from a dark chamber and do some movements back into a light chamber, but prefer to stay in the dark. And an anti-anxiety drug will increase that number of transitions and time in the light, which is our evidence that this is an anxiety-related behavior. You're familiar with the elevated plus maze, which is a beautiful assay, again, a conflict between exploring a novel area and staying in a safer enclosed area. And mice will venture out into the open arms, but prefer to stay in closed arms, and venture out more and spend more time in the open arms after treatment with an anti-anxiety drug. So these are good tests that we apply in our battery for a mouse model of autism. There's many good learning and memory tests that we use to look for potential intellectual cognitive disabilities. Novel object recognition is a good one that includes exploring two identical objects. Then there's a delay of an hour, or it could be 24 hours, or depends on what your interests are in short-term versus long-term memory. And then the mouse is given one identical object and then a new object. Mice generally choose to explore something novel over something familiar, and more time exploring novel object than the familiar is the definition of a successful novel object recognition. Object location memory is the same concept. Put two identical objects in the chamber and come back an hour or a day later, and one of the objects is now in a different location. And mice tend to explore the new location more than they would the same object in a familiar location. Also, the Morris-Water maze is a very useful task to look at learning the location of a hidden platform based on external environmental cues, or the hippocampal spatial map of the environment. And that's a good measure of learning over repeated trials. And then one can move the platform to a new location and study reversal learning, which is relevant to cognitive flexibility or insistence on sameness, that symptom of autism. We've also set up and effectively used touchscreen operant learning, visual discrimination, and also reversal and working memory tasks. So some of you have been using this touchscreen, in which the mouse just touches its nose to images that are projected on these screens to get a reinforcer. This is a very cool high-tech task that has turned out to be very flexible in terms of setting up programs that allow us to then, instead of rewarding the airplane, which has been learned, to now reward spider image to study the ability of the mouse to make a change in the rewarding properties of the stimuli. So we've now gone through so many of these mouse models, and what we have learned so far with these behavioral assays is that social deficits and repetitive behaviors do appear in some, but not all of these genetic mouse models of autism. I'd be happy to talk about the negative findings, if you like, and that associated symptoms in the cognitive and sensory and motor and anxiety-related domains appear again in some but not all genetic mouse models of autism. And one of the really interesting questions is the cases in which we do have positives, does that tell you that that gene is linked to that domain of symptom and that domain of associated symptom? And if we don't have a deficit, does that mean our assay is not sensitive enough? We haven't developed the right assay yet, or in fact, that gene is not linked to that associated symptom. So that's sort of the challenge of the field right now, is to really work out the best assays that would tell us the most about those genetic linkages. What we want to learn is how does each of these mutations contribute to each symptom of autism, so that we can develop treatments that reverse specific consequences of specific genetic mutations. So, how do you get started again? Well, what we do know is that many of these genes are involved in synapse functions in the brain. Dendritic spines are abnormal in many of the mouse models of autism and in many developmental disability models as well. Synapses are forming all the time. You're forming new symptoms right now while you're listening, or maybe doing other things while you're listening. Losing synapses, strengthening synapses, weakening synapses is going on in real time at every age. So maybe it's not too late for interventions at any age at the synaptic level to potentially treat the primary diagnostic and associated symptoms of autism based on a genetic mutation. So that's the question. Is it possible to reverse these symptoms using pharmacological therapeutics for the core diagnostic symptoms relevant to autism? So as you know, if we're looking at the synapse here between neuron 1 and neuron 2, we have many different transmitters that are released and downstream effector systems such as PI3 kinase, MAC kinase, ERK, mTOR that are activated when these synapses are forming and the transmitter is released. Luckily, the good news is that we have lots of drugs that work at these various transmitter receptors. mGluR5, AMPA, NMDA receptor drugs at glutamatergic synapses, GABA drugs for GABA inhibitory neurotransmitter receptors. We have compounds that work at serotonin receptors and its transmitter and its transporter, dopamine and its transporter, and drugs that work through oxytocin receptors and other neuropeptides. So we've got quite a good pharmacological armamentarium at the synapse and also at the downstream effector systems. We've got mTOR drugs, PI3 kinase drugs, and ERK modulators. So we've got these genetic mouse models with good assays and drugs that are commercially available or under development. Our lab and several others have proceeded to apply these preclinical tools to therapeutic discovery. And we started with a GABAergic hypothesis. There's a lot of evidence for reduced GABA inhibition being common in many different mouse models of autism and in human postmortem studies of people with autism. The GABA beta 3 subunit mutation in the GABA A receptor, the 15q11 to 13 deletion as at sites including GABA receptors. There's fewer parvalbumin positive GABA interneurons in many mouse models and in the human postmortem brain, and reduced electrophysiological inhibition has been reported both in mouse models and in human studies. So our strategy was to look at both GABA A and GABA B agonist treatments as replacements to try to restore inhibitory transmission. We've looked at mouse models in which GABA inhibition has been reported to be low. This is one set of studies by Maya Rhine, Jennifer Parrott, Maria Schultz, and Tatiana Kazdova in the lab. And the very interesting findings by Maya Rhine's work was on a GABA agonist called gaboxidol. This is the self-grooming measure that's higher in the BTBR mice than in B6 mice. And here's gaboxidol reducing the higher self-grooming in BTBR at doses of 3 milligrams per kilogram and 5 milligrams per kilogram given peritoneally acutely. It's possible that a GABA drug could be sedating. And so we looked at open field activity to control for sedation. And in fact, there was no evidence at these doses that gaboxavir was sedating. In fact, at its highest dose, it was activating a little bit in the BTBR mouse. Again, to be sure this was in fact a reproducible finding, Maya ran cohort 2, and again, here's more grooming in BTBR than B6, and again, the 3 and 5 milligram per dose reducing self-grooming in B6 and also reducing self-grooming in BTBR with no effect in the open field activity measures. And Maya, who's an extremely careful scientist, then ran a third cohort and again saw less self-grooming in BTBR after treatment. And again, no evidence for locomotion being inhibited. So that's pretty strong corroborative findings across 3 cohorts that this GABA-A agonist may be effective in the repetitive behavior domain in at least one mouse model of autism. For the GABA-B receptor, we looked at agonists called R-baclofen and looked at 2 different mouse models that show repetitive behaviors and social deficits, at least in one. And R-baclofen reversed the absence of sociability in the BTBR mouse. Here's normal sociability on the 3-chamber task in B6 mice and lack of sociability in the BTBR mouse that is restored after the R-baclofen treatment at doses of 1 and 3 milligrams per kilogram. That's on the chamber time parameter. Same thing on the sniffing time parameter. And this control measure of general activity entries between chambers was not affected by R-baclofen at these doses. In terms of the grooming scores, again, here's more grooming in BTBR than B6, and R-baclofen did reduce self-grooming in the BTBR mouse, as well as a second repetitive behavior task, digging in the litter and covering up marbles that were placed on the surface of the litter. And then here's a second inbred strain, C57J, that engages in stereotyped vertical jumping, just jumping up and down in the cage. And that high stereotype jumping was also reduced by the same doses of R-baclofen. So to my amazement, we have both reductions in the social domain and in the repetitive domain with this GABA-B agonist. The activity levels, again, could have been affected in terms of sedation with the GABA drug, but in fact, the doses used for R-baclofen had no significant effect on distance or horizontal activity, did reduce center time a little bit in B6, but no significant effects in BTBR, did reduce total distance and activity in center time a little bit in C58, but just at the higher dose. So this indicates that we may have a somewhat narrow therapeutic window in which R-baclofen has beneficial effects without having sedating effects, and that's important to think about in terms of clinical studies. We then went on to look at the 16p11.2 deletion mouse. This is a syndrome in which some people with that deletion show the symptoms of autism. Others show areas of intellectual disability. And there's evidence for MAP kinase genetic mutation within that deletion range, indicating that GABA reduction could be an issue with this line also. So Tatiana Kozdova and Melanie Schaffler in the lab looked at behaviors relevant to autism in the 16p11.2 deletion mouse. This is a collaboration with Mark Baer's group at MIT. His team, Laura Stoppel and Dr. Heinen, looked at several different cognitive tasks and found improvement with R-baclofen. And we did the same. We looked at the object location memory task. And here's good object location memory in the wild type littermates and absence of object location memory in the 16p deletion heterozygotes. They showed same amount of time exploring the object in the original place and the new place. And here's R-baclofen restoring object location memory in the 16p heterozygotes. In terms of social behavior, this is R-baclofen administered in the drinking water for 2 weeks. This is a chronic study. Here's the 16p11 pets showing less nose to anogenital sniffing as compared to their wild type litter mates, and that was restored after the arbaclofen treatment. And same thing in terms of sniffing duration and number of bouts of sniffing. So these are preliminary indications that these compounds might be useful. There's a lot of issues in the animal data that we need to think about in terms of dose, in terms of which symptoms are improved and which are not. But I wanted to share with you that arbaclofen is currently in a clinical trial for autism spectrum disorder through the EU AIMS consortium. These are ongoing trials. I think we've got 2 or 3 more years before we know what the controlled treatments are looking like and the effectiveness of arbaclofen in this population using a variety of new outcome measures. It's a long road to effective therapeutics. There's a lot to be considered in terms of what are the right outcome measures, what are the right groups to use, what are the right subgroups to look at, what are the most effective doses, when to start the treatment. Neurodevelopmental disorders really is at the beginning of trying to design the proper experimental approaches to look for therapeutic interventions. So I do want to mention to anybody who has a child with autism or knows somebody who has a child with autism or knows an adult with autism, behavioral interventions are still the standard of care right now. They're very effective for treating multiple symptoms of autism. So if you have a situation in which the child has not entered a behavioral intervention yet, start as early as possible. Age 2, at very first diagnosis, there's extremely good behavioral interventions that are fairly effective in bringing the child up to a better standard of lifestyle. But in terms of therapeutic interventions, there are many compelling hypotheses about pharmacological interventions to pursue. These are being tested in a variety of mouse models of autism by our lab and by many other labs. Each of these has its own distinct pharmacological and behavioral abnormalities in the mouse model and in a therapeutic intervention. And we need to focus on these subgroups in an attempt to develop precision medicines that address the subgroups of people, such as people with GABA deficits, with autism spectrum disorder, as well as other neurodevelopmental disorders. I'm afraid that using very broad strokes will not get us there because people without GABA abnormalities probably won't respond to GABAergic drugs, for example, and the same for other therapeutic targets. And I think companies and clinical researchers have understood this point, and there's several very interesting studies being planned or in progress in the clinical domain. So we're cautiously optimistic at this point and hope that some of you might be interested in joining this fascinating search for therapeutics for autism. It's an honor to be a member of IBANGS, which is the premier society for behavioral genetics in our neuroscience community. And really grateful for the opportunity to give this talk today and to thank the many of our lab members and collaborators and funding agencies who supported this work. And here's just a few of the people who've been in the lab recently who contributed some of the studies that I've talked about today. So thank you very much, and I look forward to your questions.
Thank you, Dr. Crawley. I'm going to clap on everybody's behalf. In the days of Zoom, you missed the audio clap. So I'm bringing it back. At the very end of the talk, you asked about, you were talking about sort of broad strokes, heterogeneity of the disorder, and John Crabb actually had a question related to that. So you said that we wouldn't be able to use broad strokes, but what kind of I guess in broad strokes, what kind of heterogeneity or what is known about the heterogeneity or subtypes of human autism, like from a clinical perspective, like what are all the subtypes out there? And if we do have like discrete subtypes, can we use Can we use genetic mouse models to map those specific subtypes as opposed to, you know, autism as a whole? Because I think that's one of the, I think, and Dr. Crabb knows that that's one of the sort of tough nuts to crack, is when you have something that's so heterogeneous in human populations, how do you actually map that in animals? And the answer is you can't without having more specific measures. Can you elaborate on that a little bit?
Sure, it's a wonderful question, and thank you, John. To me, this could be a whole other lecture, and I could give you many, many different examples. To me, one of the most interesting ones is intellectual disabilities that are associated with autism in some people, but not others. So there's some people with autism who have very high IQs, remarkably, some really in the genius range. And there used to be a diagnosis called Asperger's syndrome, in which the intellectual disability was not a component. Asperger's got folded into the broad spectrum of autism in the DSM-5 revisions. So we don't really talk about Asperger's, but I think that's one of the most interesting subgroups that, in fact, to me, one of the fascinating questions is whether social deficits are in fact a kind of social intellectual disability, inability to interpret cues, to process social information quickly in real time. That might be a problem of intellectual capacity that feeds into people's general intellectual disability. But there are these very smart people who also have social problems. And I think if we could focus on that subgroup, we might really get a better set of insights into what the social problems are as distinct from all of the cognitive disabilities that might impair individuals from learning about social cues, and the sensory problems and the motor problems and the anxiety-related problems that might prevent them from engaging in social behaviors. Another point I'll just mention quickly is that the comorbid disorders such as Rett syndrome or Angelman syndrome or tuberous sclerosis have major physical problems, such as Rett has motor degeneration, Angelman's has motor problems, and tuberous sclerosis is a seizure disorder primarily. If one could factor out those components and the genes that are associated with the seizure component, you might again be able to separate out the more behavioral versus the more physiological underlying syndromes associated with that genetic mutation. So that's a start.
Thank you. Thank you very much. For the next question, I'm going to ask Dr. Kathy Fernandez to ask her question. Go ahead, Dr. Fernandez.
Thank you, Paul. Thanks, Jackie. Fantastic talk. Hello, everyone from London, rather dark one. Jackie, it's kind of, hi, Jackie. It's a question really I often get with clinicians, because as you know, I'm sure you've had these questions before. There are real challenges to using animal models to look at behavior from clinicians. And the frequent claim they make is that animals don't have sophisticated behavior. It's fine to do everything else in them, but not to use them for behavior. So of course, I know you would probably disagree as I do. So I think the challenge is really that they do have sophisticated behaviors. We're just not very good at measuring them yet. So I just wondered what's your view in perhaps the future for behavioral experiments, maybe more sophisticated ways to look at social behavior, and particularly olfactory communication, which we don't have very sophisticated tests yet for.
Very good points, Kathy. And as many of you know, Kathy is one of the leaders in mouse behaviors relevant to autism and other neuropsychiatric and neurodevelopmental disorders. So you're absolutely right. We don't have the full armamentarium of measurements of mouse behaviors, and we just have to keep being smart and creative and come up with better and better assays. And I would encourage the younger people on the call to think about this and start by just watching your mice, watching them in different situations, setting up environments that are more complex than just an empty open field, thinking about social groupings rather than just dyads of two mice together, thinking about delivering olfactory stimuli in different creative ways. I think you're absolutely right that we get stuck on a few assays that have been working well and they're simple and easily quantified. That's perfectly legitimate. And certainly one can use the standard assays in ways that can tell you a lot about brain function, correlations to neurophysiological and neuroanatomical abnormalities. In terms of trying to relate the animal behavior to the human behavior, I just try to be really careful and always say these are face validity to analogies of the human symptoms. They are not the human symptoms. This is what a mouse model can do for you, and this is what a mouse model can't do. And don't oversell the correlation between human and mouse behavior too strongly. And usually that's acceptable and shows people that you can make certain statements, but not others. You can say that we're looking at a sensory-based task that in mice is olfactory and human might be visual. And in mice might look at the olfactory cortex or the olfactory neurotransmission genes and pharmacology, and in humans the visual system is a different set. But at some level you're getting into higher integration of sensory and cognitive processes, and that may be the same for mice as for humans. And so you just have to be really careful about what we know and what we understand as our limitations.
Thank you, Jackie. Thanks, Jackie.
Thank you. Another question from Lara Montana. Her question, oops, sorry, I lost it. Her question is, first she says, great talk. She said that you mentioned that overinterpretation of the social interaction assays is a problem, and that these were designed to look at within subject differences, not necessarily across subjects or strains. So how is this better done to be able to look at social interaction across strains? Like she's suggesting something like a social index, like you mentioned, but maybe there's some accepted way that we don't know about. She's asking because she's doing behavior phenotyping across many different strains, basically a genetic screen using different backgrounds, different strains.
Thank you. That's an important question. And again, I'll just mention that this three-chambered social task has a lot of variability. If you test the same mice again next week, you'll get very different numbers out for each strain or for each treatment group or for each genetic mutation line. And that's why we don't compare actual absolute values of number of seconds spent with the novel mouse across strains or across treatment groups or across genotypes. It's just not sensitive enough. That's the simple answer. So if you really do want to look at quantitative parameters, I would say don't use the three-chamber task, but use reciprocal social interaction of freely moving mice. Using an index for the three-chamber task obscures the activity component. So you might have mice with very low activity that make very few movements into the chamber with a novel mouse. They make one more than two is a 50% increase, but that's not really biologically an important difference. So you need, and if they're doing very high levels of activity and they make 90 movements into the chamber with a novel mouse and spend a lot of time there, but they're actually running around in the chamber, you'll again get a false positive comparing across strains. So I think if you're interested in quantitative studies, you really want to use a quantitative assay. And it's more laborious, and maybe you have cleverer ways to look at individual activity and social engagement in freely moving mice that will speed things up a little bit. But again, I just want to emphasize that the three-chamber test really can't give you that kind of quantitative information.
Thank you very much. I realize now that I might have said his name backwards.
Problem.
Hi.
just a comma. My apologies. I managed to embarrass myself. So it's only a matter of time for that happens. I've got some questions about specific assays. So I'm going to do a double header. One from Hannah Wider. Thanks for a great talk. Has your lab ever looked at nesting or spreading? And
I'm
then,
sorry, I'm having a little trouble hearing. Could you say that again, please, Paul? Has our lab ever looked at
nesting behavior or nestlet spreading ever in different disease types?
Yes, that's a wonderful question. It's one of our first collaborations with Tony Winshaw-Boris when he was at NIH, was with a disheveled one mouse that didn't form nests and didn't sleep in huddles. And that was a very interesting phenotype. They just didn't fluff up the nestlet. And they slept in different parts of the cage surface. And we interpreted that as a social deficit. In fact, a lot of people since have started scoring nest building as a parameter in their phenotypic and mouse models of autism. And I'm not convinced that nest building itself has a social function. As you probably, many of you know, mice build nests in the real world as protection from predators and to conserve body heat. And they sleep in huddles inside the nest, again, for camouflaging and for staying warm. I think nest building is probably a housekeeping behavior, not a social behavior. Scoring the animals sleeping together in a huddle inside the nest might be a measure of sociability. If an animal's excluded from the nest or chooses not to join the others, the time spent in the nest together, I think, might be a valid parameter. But I would say that nest building should probably be excluded from phenotyping for social behaviors in mouse models of autism. I'm not sure if that's exactly the question that was asked, but maybe you want to elaborate?
Yeah, that was exactly, that answered it really well. Thank you so much. I've always wondered about that one. I feel like it's one of those things thrown around to be a way to look at certain behavior for a bunch of different disorders or animal disorders, and I was on it there.
Thank you, Hannah.
The cell phone too close to your microphone? It's better now.
Okay.
I don't know if you've done anything, but that sounds better.
Oops.
Yeah, that sounds great now.
Okay, good.
Okay, another question about specific assays. Julie Blendy saying, thanks for a great talk. Could you give your opinion on the value of the marble bearing test in terms of anxiety versus compulsive behavior, because they're used for both when assaying autism spectrum disorder like behavior?
Sure. Thank you, Julie. Great question. Great to see you and hear from you. So marble bearing turns out to be a repetitive behavior, not an anxiety-related behavior. This was worked out very well by the late, great Rich Paler, who really spent a lot of time looking at marble bearing. And our lab has also, and what we see if you watch the mice is that what they're really doing is digging in the litter around the object. And this is something that mice just do all the time. They dig and dig and dig in deep litter. And I'm not even sure that they're targeting the marble. I think they might just be kicking up so much litter that the marble eventually gets buried. And as they move around the environment, they're kicking up lots of litter. So you use fairly deep litter, like 2 inches deep, 5 centimeters or more deep. And over time, you can see the litter ends up in these sort of hills and valleys across the surface of the cage that might be used to bury the marbles. And I think it might just be by chance that they're burying so many marbles. I might be wrong about this. Some of you I'd like to hear your comments. But it seems like the marble bearing is just a repetitive digging phenomenon, much more than something related to anxiety. I know it's been in literature early on as used for anxiety-like tests, but I don't really think that we've seen any evidence for that. That's a good question. Thank you.
Thank you, Jackie. Appreciate it.
That is a good question. I think a lot of models like that are based on predictive validity sort of have that circular logic where you find anti-anxiolytic drugs and you say, therefore, this is a model of anxiety, which I think is problematic, as you pointed out. From Anna, Anna's asking if you've ever looked at neurogenesis in your models.
Oh, that's a fascinating field, Anna. We haven't ourselves, but I'd love to talk to you more afterwards.
And Carl Clark is asking how often human variants in these genes are resulting from complete loss of function, like a mouse knockout.
Right, thank you, Carl. That's a very important point. So most of the human mutations are heterozygous, and a lot of the mouse models, if you have the mutation as a null deletion, the mice don't survive. So a lot of the mouse models also are heterozygous. Not all though. So for example, Shank3B mice that we've looked at have a more severe phenotype when it's a null than when it's a heterozygous. Whereas the human syndrome appears only as hets, as one allele is still present. So it's not a one-to-one correspondence that all of the human disorders are heterozygous. Some of them are null mutations or complete loss of function, and some are not. And the same thing for the mouse models. So it's sort of a case-by-case basis to make sure that your genetics of the mouse model matches the genetics of the human disorder, and that the symptoms emitted by the mouse with that mutation appear in the hets if it's a primarily human heterozygous disorder. And that doesn't always turn out to be the case. Sometimes the hets in mice show no phenotype at all, but sometimes it's a pretty good, robust phenotype.
I'd also guess there's a range of alleles in the human patients, so where they're not like the specific alleles, it may be a complete loss of function, but sort of a hypomorph or something. Is that probably accurate, or?
Yes, that's exactly right. The big range across genetic mutations in autism.
So since we're talking about the nature of the mutations, something that I've always been kind of about with autism, de novo mutations. Like you have such, I mean, as you know, autism is really heritable, and a lot, and you've identified and investigated so many different variants in mice. But in humans, a lot of these mutations are happening de novo. And I just sort of like, I want to understand the cause of that, the cause of the actual mutations happening in the first place. Like what is underlying the potential vulnerability to actually get a de novo mutation in the first place? Because that's not something that's inherited necessarily, or is it? Is there a genetic vulnerability factor to getting de novo mutations throughout the genome?
Right, you're right. There's genetic vulnerability to getting more mutations in general. But if you just look at the number of mutations that people have who seem to be normal versus number of mutations that turn out to be de novo in people with autism, it's probably the normal rate of mutations. As you say, de novo mutations means, by definition, that they occur randomly and just happen to be on a gene that affects synaptic development or neuroanatomical development or brain function in some way, leading to a brain disorder. And as you say, the number of genes in which there's been de novo mutations linked to autism, associated with autism, is very high. And each each of those genes has a fairly small number of people with that particular mutation. Like maybe 20 people have been identified with a given mutation, or 100, but not thousands and millions.
I see.
So it's A, random, and B, de novo. And I gave a talk at Cold Spring Harbor many years ago, very similar to this one. And Jim Watson was in the audience, and he said that what you're describing is exactly consistent with random gene mutations. It has nothing special about autism or about the brain function. So, and other people have found the same thing, that it's just bad luck, just the way mutations work in general. There is some evidence, as I mentioned earlier, for older parental age be a risk factor. And so possibly, just accumulations varying across the lifespan of the parent, including spermatogenesis factors, could be contributing more general de novo mutations over time. And maybe we'll find other factors like that increase the number of de novo risk factors for autism. But it's probably the same as environmental factors that increase the risk for various other disorders the same way. Or at least that's my reading of the literature at this point.
That's really helpful. Thank you. I think if you'll bear with us, we'll squeeze out 2 more questions, if that's okay. One from Susan Maloney about the going back to the marble bearing task. She's seen it as interpreted as neophobia. Do you think that that's a possibility, or do you think it really reflects if it's only repetitive digging?
Yeah, could be, Susan. And I really haven't studied marble bearing in the depth that you probably have. And so I don't have any further insights than what I've just mentioned in response to the earlier question. Neophobia is certainly something that mice demonstrate when anything new is put into their cage. Usually the marble bearing task is set up in a new environment with new, fresh, clean litter and marbles. So there's a lot of new factors in the environment. And maybe the marbles are particularly just noticeable, and mice are responding more specifically to the marbles than to other things. Those are interesting questions that maybe some student would like to take on as an experiment and try to tease out whether there are very specific factors about marbles that mice notice and want to and tend to bury more than other objects or other novel factors of the environment. It's a good question that I don't have an answer to, but if somebody would like to get into this, I'd be happy to talk with you more afterwards.
Thank you, Jackie. That was really informative. I've seen it in a couple of papers as decreased marble bearing interpreted as neophobia, and I haven't dived into exploring or validating that yet, but I think it would be something worth doing in the future. I've had a handful of models that have shown decreases in bearing in this task, and so it's hard to interpret if I also see anxiety-related phenotypes in other tasks. So exploring it as neophobia might be something interesting to dive into in the future. And thank you also for a great talk.
Great, thank you for your good questions, Susan.
Okay, one more question. This is, I'm going to call on Philip Cosell to ask his question. Sorry, Philip, if I mispronounced your name. Go ahead.
Sorry, that's not a problem. That was actually correct.
Oh,
Jacqueline, I appreciate, I hope you can all hear me clearly, but
yes.
I really appreciate this talk. I actually work with a mouse model of, I'm going to say Alzheimer's disease, although it's really amyloid beta related neurodegeneration. And I've used actually a few of your assays that you've developed over the years in testing sort of social behavioral aspects in these mice. I just had a really quick question with regards to the specific, like the 3-chamber task and the reciprocal social interaction task. in that, how much of a role do you think may be played by social anxiety? And B, is there a way to differentiate, for example, social anxiety over I'm going to call it a simple social disinterest?
Yeah, you're asking tough questions, and I don't have the answer to them. So social anxiety, social phobia, schizophrenia like social avoidance. These are all very important for human disorders. So the social problems that you might see in depression look very different from those you see in schizophrenia, which reflect paranoia, look very different from those you see in autism, look very different from the social problems you see in people with Angelman syndrome, which are actually very happy often and engaged people. It's a challenge to think about how to look at these subsets of social problems in mice. Mice have a social repertoire. As we talked about earlier with Kathy Fernandez, it may be a lot more limited than humans, but perhaps we're not using the right assays. I wish I could answer your question. I don't have any brilliant insights to offer, but this would be fun for us to talk about. Anybody wants to stay on the call or get in touch with me afterwards? Thank you.
Okay, thank you everyone for all your great questions and for being so engaged with Dr. Crawley's talk. And thank you, Dr. Crawley. That was fantastic, as usual, as expected. Thank you.
Thank you, Jackie. Wonderful.
Thanks, Jackie. Bye.
Thanks everyone for attending.
Happy holidays.
Yeah, happy holidays, everyone.
Stay safe and healthy.
That was the hardest question and answer session that I've moderated. So I think that's a good thing. I was getting a lot of questions, so that was good. That was great.
Wonderful. I'm just looking at them now. Yeah.